going to start off with a reminder, and then I do have a little review slide for you. So the research article report is due a week from today. Print that in on Blackboard, please. As always, if you have any questions, let me know. For your warm-up, I want you to try to work through the different steps of cellular respiration. So I popped this figure up here for you. I put this on the screen last class as well. So this is kind of highlighting the different steps of cellular respiration. Of course, glycolysis, or pyruvate oxidation, which is what I call the step 1.5, citric acid or Krebs cycle, and then oxidative phosphorylation. So lay out some of the main steps in more simplified language in a way that makes sense to you. Do this with a partner for a little bit. You can use your notes, like I said. So try to work through this, review what we went over last time before we do some more kind of heavy lifting today. Okay, ready to go over this a little bit together? All right, cool. Glycolysis. So in this, I also say let's focus on some of the inputs and the outputs. So let's kind of do that as well. Glycolysis, what's the deal here? What goes in? Glucose, cool. Yeah, what else do we put in? Got to give it some energy. Get going. Yep, 2 ATP go in. Lovely. We break that glucose molecule down into 2 other carbon-based molecules, right? We get those G3P, and then those are converted into pyruvate, right? Okay. We also get what out of that process of glycolysis? What's produced? Overall, 2 ATP. Yep, cool. So our pyruvate molecules that are produced in this first step of glycolysis are going to go on to that pyruvate oxidation if oxygen is present. Remember, if we have oxygen, this will occur. It will eventually be converted to acetyl-CoA through that oxidation reaction. All right, next up, citric acid cycle. What happens there? What goes in? Thing we ended with here. What goes into the citric acid cycle? Acetyl-CoA, yes, cool. So that goes into our citric acid cycle. And when that is occurring, we end up getting some lovely electron-carrying molecules, right? We get NADH, we get FADH2 in that process. We also get CO2 and ATP as a result of the citric acid cycle. Then those electron transport molecules, or those electron carrier molecules highlighted here, are going to come over and participate in our electron transport chain and ultimately amnioxymosis, which is what I have highlighted again here. This is also a slide I put up last class. Again, I wanted to kind of make sure we're re-going over this. Electron transport chains are very important to living organisms because they help fuel these metabolic processes. Electron transport chains do not just occur in cellular respiration. We're going to talk about where they occur in plant cells today. So they're a certain type of process, but they exist in more than just the kind of talked about. So remember here we have these electron carrying molecules, and this figure is lovely. It kind of annotates this carrying electrons from food. So it's really getting at this bigger picture here from where this energy is originating from. These electrons are going to be transported through this series of different protein complexes. Protein complexes are kind of groups of proteins put together. It's going to go through this protein complex. As that's happening, protons are being pushed out into this inner membrane space. So these H positive circles, those are protons, those hydrogen ions. So those are getting pushed out, and then eventually ATP synthase is going to allow those hydrogen molecules, those protons, to flow back through it. Now that is in accordance with that electrochemical gradient, right? Because we have a higher concentration out here in that inner membrane space relative to the inner mitochondrial membrane. So it's going to follow that electrochemical gradient, and ATP synthase can harness that energy and produce ATP by churning a little molecular rotor. So that's what's happening here in that chemiosmosis. This produces a lot of energy, a lot of ATP. This is where we get the majority of ATP, is in this oxidative phosphorylation step. Good? Okay, if you need to keep reviewing this, please do. Again, you can read the textbook, you can watch, there's so many videos online that have visualizations of this, figures of this, so you can watch those as well. You can watch my lectures over again. So do please review this. It is complicated. It's something that's important, so try to look over this. Going forward a little bit, I'm kind of finishing up our lecture on cellular respiration, how many ATP are made from a single glucose molecule. We can kind of keep track here and do the math. We get 2 from glycolysis, which we were talking about at our review. We get 2 from the Krebs cycle, and then a majority of our ATP produced, as we were just talking about, via ATP synthase during oxidative phosphorylation. The total brings us to about 30 or 32. However, this estimation fluctuates based on a variety of different factors. ATP yields can vary based on which specific shuttling proteins are involved in the electron transport chain. In different organisms, in different cells, those protein complexes vary. So some just happen to be more efficient with that process. Interestingly, we also know that the ratio of NADH molecules to ATP produced is not 1-to-1. There's like half involved there. So that's also why we get fluctuations in our estimations sometimes. It's not a perfect 1-to-1 ratio, so we can't get perfect numbers. When we talk about this process overall, about 34% of the chemical energy available in glucose does end up getting converted to ATP. So we consider this to be an efficient process. Cellular respiration is fairly efficient. There are specific environmental conditions that can be adjusted. So different organisms might have reasons to want to slow this process down or speed this process up. So the rate of cellular respiration can fluctuate as well. A good example of why an organism might want to slow down its rate of cellular respiration is if you think about hibernating mammals, for example. They're going to be sleeping during the winter. They're not going to be moving around and needing a ton of active usable energy. So in fact, if the cells are producing a bunch of ATP, they're not going to be able to use it, which is not ideal. That's actually fairly problematic. So there's certain chemical and cellular mechanisms that allow those types of organisms, hibernating organisms, to slow this process down and adjust what is being converted where. So they can actually convert some of that energy from food or fat into heat, which allows them to stay warm. So that's what this last bullet point is kind of talking about here. Energy from glucose that is not converted to ATP can also be used as heat. We also know that energy is lost as heat when organisms are kind of moving through their environment with these conversions. As promised, I'm not going to make you learn the names of all the different enzymes involved in this process. That's kind of something that used to occur in lots of biology classes. I'm not sure if people in our department are still doing it as much, but you'll notice if you focus in on some of the names here, these long, kind of hard looking to pronounce names, those are mostly all different enzymes. So we have like phosphofructokinase, which is a really important enzyme here. Lots of different ones. if we look over here, the cytochrome reductase. So there's enzymes involved in each of the different steps of cellular respiration. So never forget, enzymes truly rule the world. Just a reminder. I did mention that we were going to talk just very briefly about what happens if there's no oxygen. So remember that glycolysis step can occur. That's happening in the cytoplasm. And then if oxygen is present, those molecules are going to enter the mitochondria. If oxygen is not present, then cells can do lactic acid fermentation, which produces a lot less ATP than cellular respiration in the presence of oxygen. But if you do not have oxygen, this is something that can still be helpful to produce that usable cellular energy. As I have laid out here, this produces only 2 ATP, and you also get lactate as a byproduct, which is often known as lactic acid. Lactic acid gets a bad reputation. People associate it with muscle soreness. That's actually not true at all. So lactic acid is not the reason you get sore after a workout. Now, your muscle cells do tend to do a fair bit of lactic acid fermentation. They have to do this anaerobic process, and that's why we get a buildup of lactic acid in tissues that are being used during physical exertions. However, the things that make your muscles sore are actually like small micro tears in the tissue, which causes swelling and inflammation. So lactic acid does have to get broken down in the liver, but it's not causing you to be sore. This process of lactic acid fermentation occurs in the cytoplasm, so this doesn't happen in the mitochondria. And this figure is kind of showing that, and it's kind of a weird figure, but you can see we're starting here with glucose. That first step of glycolysis occurs. We get those 2 pyruvate molecules. Then through the process of fermentation, we get these 2 lactate molecules. So that's what this figure is kind of talking about here. Questions? Okay. As I was kind of talking about the rates of cellular respiration, this is another important note about cellular respiration. We know, of course, we consume things other than just carbohydrates, right? So polysaccharides and disaccharides are both types of complex carbohydrates. So those do have to get broken down before those can be used in this process, of course. We do also eat things like lipids and proteins. Those can also be used to make ATP. So it's not like glucose is the only usable molecule. There are other molecules that can be important for making ATP. They do have to go through steps of being broken down into their specific components. So lipids, for example, have to get broken down into their fatty acid chains and then their smaller molecules. Same deal with proteins. They're going to get broken down to their amino acid monomers. And then those amino acids actually have to get broken down even further before they can be involved in this process. So lipids and proteins don't directly contribute to like that first starting bit of splitting a glucose molecule because they're not glucose molecules, but they serve as intermediaries in other steps of cellular respiration. So that's what I'm talking about here. Amino acids have to lose that nitrate group before they can be used. But once they do that, they can help out with the process of cellular respiration and help contribute to producing ATP. Cellular respiration can also be regulated. Mostly this happens by this enzyme phosphofructokinase, which is important in that first step of glycolysis. Something that's kind of interesting, cells and organisms are not wasteful, so they're not going to produce more molecules than what they need. So there's certain regulatory mechanisms like this phosphofructokinase enzyme. It essentially is involved in a process that allows the cells to sense how much ATP is present. And if there's a lot of ATP, phosphofructokinase is going to slow the rate of cellular respiration down. And if there's not very much ATP that's sensed, it's going to kind of speed that rate up. So if you're doing a lot of physical activity, you need more ATP. If you're doing less activity, you need less ATP. And so organisms are able to kind of adapt to that. As promised, we're going to talk about why cyanide killed the folks who were taking the cyanide-laced Tylenol. Hopefully this has not been haunting you since Monday, but we remember there was a safety seal, there was the Tylenol that was causing all these troublesome deaths. Why does cyanide kill? It is a known problem. You might have heard about cyanide before. This has to do with cellular respiration. So the specific mechanism for why cyanide is lethal is because it inhibits the electron transport chain. And that's what I have a figure here for you. So cyanide binds to cytochrome C oxidase, which I've highlighted right here, which is one of the proteins that's in the electron transport chain. So it doesn't allow the electrons to be transported in that transport chain. Therefore, what's not going to occur? If we don't have the electron transport chain working properly, what's not going to happen? We're not going to get a whole bunch of ATP. Yeah, that's not going to be happening. That proton pumping out is not going to be working effectively. So it shuts down our ATP generation, which is super problematic because then cells tend to die if they do not have the necessary energy. This next question I have up here for you. Survivors of cyanide poisoning can get Parkinson's disease or other neurological disorders. Why do you think that is? And you can talk about this with someone. Parkinson's disease is a central nervous system disorder, if you're not familiar. So we can get these neurological conditions once there has been cyanide poisoning if you happen to survive. Why do you think that is, Charles? Why does that happen? Okay, what are you thinking here? What's the deal with the neurological disorders occurring after cyanide poisoning? Sure, yeah, why?
The cyanide damaged the cells of the nervous system, and nervous system cells typically they don't reproduce. You have about the same since birth, unlike most of the cells in the body that will reproduce and fix themselves. The neurons will stay mostly damaged, which will lead to psychological disorders down the line.
Yeah, neurological disorders. Totally agree that neurons do not regenerate. So yes, there's lots of cells in the body. Some of them grow quickly and reproduce quickly. Neurons definitely aren't one of them. I do agree with that. Does anyone else have any other thoughts here? What damages the neurons? What causes that problem in the first place? I was telling you a while ago, your brain uses a lot of energy, right? It needs a lot all the time. So if it's not having that constant influx of ATP, that's what causes ultimately that cellular death. So that's what can lead to those neurological problems. So kind of interesting. Cellular respiration, it's out here doing things.
How long do you get Parkinson's?
That's a good question. Probably depends on how severe like the poisoning is. If it was super severe, you probably would not live to experience that. And I'm not sure what the onset is. It's a great question. I should look into that. Any other questions? Okay. I do have this little activity for you, and I am curious about this. I want you to do this. What is one question you still have? Cellular respiration. Trade questions with partners, see if they can answer it for you. So think about it. You can look back at your notes from last time as well. If your partner can't answer your question, you can email it to me. I will answer your question. So think about what you still want to know about cellular respiration. See if someone near you can answer this question.
So
I'm pretty sure he's already
Okay. So again, if your partner could not answer your question, send it to me. I will answer them. I'm not going to answer them right now. I do want to get to photosynthesis, but send me a question if you want me to answer it, and I will. One other thing before we move on, I do want to note, I was talking with a student today, and not a student from this class, a different student, and I was like, they were saying, what are you doing in your Gen Bio classes? Oh, we just finished talking about cellular respiration. They're a senior in the department, and I said, what is the point of cellular respiration? I say, it makes energy. Not entirely truthful, so I just want to make sure that we're all super clear about what cellular respiration does. It makes usable energy, right? So we have glucose. Glucose is an energetic molecule. The sun provides that energy. So the body is not making energy. Mitochondria don't make the energy, right? So we have to be clear here about some of those distinctions. It's taking one form of energy and converting it into a usable form. So we're not really making energy, just so we're clear. All right. We're going to go into photosynthesis today. This video is a little bit longer than the ones I tend to link. I think it's like 12 minutes, maybe 15. It's not super about photosynthesis, but it is about plants and pollination and the interesting nature filmmaking in the Everglades. So feel free to check that out. It's great. It's about ghost orchids. I also put a note here. I recommend reading more about this process in the book. If you are getting a little lost, photosynthesis is super duper complicated. Honestly, we're doing a pretty brief overview of this process. If you go on to take botany, you'll once again talk about photosynthesis, probably in a little bit more detail. Again, this process is very complicated. I am simplifying it to some extent today. Sure, yeah.
Would you be able to post
Should be posted now. Let's go take a look. Maybe it's hidden. Hopefully I don't hit the duo. Yes, it's hidden. Sorry, that's the automated setting. All right, should be visible now. So if you want to follow along, you sure can. Go ahead and update your Blackboard or refresh it. And there you go. All right, back again. Learning objectives for today. I want you to know how photosynthesis contributes to the biosphere. We're going to talk about inputs and outputs for the two portions of photosynthesis. We break it down into two steps, light reactions and then dark reactions, which is light independent reactions. Sometimes they're called that. I want you to understand how carbon is related to plants, photosynthesis, and the environment, which hopefully we'll have time to talk about. And then we're going to talk a little bit about trophic levels very briefly. So what's goals today? Getting started, I put this figure up here for you last time. It showed both photosynthesis and cellular respiration. However, today I've highlighted that photosynthesis portion of this process. So we can see the sun up here at the top emitting this lovely light energy, which those yellow flowers and those plants are able to absorb and convert into these organic molecules, which those are useful for other organisms in cellular respiration. Now, that sun energy gets converted into sugar. Sugar is food. We kind of talked a fair bit about this. Plants are what we call primary producers because they are able to use that light energy and then convert inorganic molecules, things like CO2, into organic molecules like sugar-based molecules or carbon-based molecules that can be used. Not to be over dramatic, the entire planet depends on plants. So their ability to do this is really, really, really important to the functioning of the entire biosphere because all other organisms are going to be able to take advantage of their stored sugars. Plants and photosynthetic organisms are the most abundant primary producers on the planet. And sometimes the species that are able to benefit from this process, they're doing so directly. If they are an herbivore, directly eating the plant, it might be an indirect benefit if it's something like a human coming along eating a cow that has maybe been foraging on plants. So this could be a direct or an indirect benefit. Okay. Questions here? Okay. So flowering plants are good eats. Lots of things like to eat them. As I've said many times, plants can capture this solar energy. They convert that to chemical compounds. As a result, they're considered autotrophs. If we break this word down, auto means self. They're able to make their own food. So autotrophs are things that can make their own food, and plants we consider to be photoautotrophs. And those are organisms that use light to make their own food. Other things that can make their own food are like bacteria. Occasionally they can do that as well. Now, heterotrophs are organisms that have to eat other things in order to get energy. So we would be considered heterotrophs. They're organisms that cannot produce their own food. And this is a fun example. Does anyone recognize this? I don't know what this is. You can see it in Kansas sometimes. Pretty big, actually. Scale. Tomato plants. Have you seen these? Tomato hornworms. Did I hear that? Because that's what they're commonly called. Yeah. So this is a specific type of hummingbird moth larva. They do turn into hummingbird moths or sphinx moths later, but yeah, as larvae, they love to eat certain kinds of crops, and they do get pretty big. And this example, you can see a kestrel feeding its baby kestrel, a lovely hawk moth larva, and an article in the Audubon, actually, where I got this picture, they called these larvae the hot dogs of the animal world, which is kind of hilarious. They call them nature's hot dogs because they're such good eats as a result of them being able to take advantage of all those awesome organic molecules in plants. This kind of relates us to the trophic levels or trophic pyramids that we could talk about the way different organisms essentially interact. So here we have both a marine ecosystem and then we have a terrestrial system. You'll notice some key differences here. This marine system is showing decomposers, which are playing very important roles in terrestrial ecosystems as well. They're just not featured on this different trophic pyramid. Now, the base of the pyramid in both of these marine and terrestrial systems are comprised of these primary producers. That really helps underscore how important plants are to the functioning of ecosystems. And we have, again, talked a little bit about this. The metabolism is something that we've gone over before. That is, of course, all the breaking down and forming of molecules that happens inside cells. Generally, breaking molecules down releases usable energy. Then we can build different molecules. We haven't talked about bioenergetics before, which is how energy is flowing through systems. That's a specific word for it. So energy flows, and that is the bioenergetics. Energy can be transferred. We did talk about the laws of thermodynamics a little bit, so related to that as well. Okay, getting into mostly new stuff here now. So this is showing the different types of waves. This is the electromagnetic spectrum. And different types of light are differentiated using the electromagnetic spectrum. So this figure is kind of helpful in that it's showing these different wavelengths based on, or comparing them to different things you might be able to kind of use for scale. So we have really big waves, things like radio waves are really, really big. And then we can get down to gamma rays, which are really, really small. Smaller waves transmit more energy. So that's a general rule. And over half of the energy that the sun emits is infrared, and then 47% is visible light. So the sun is producing mostly infrared and visible light, which is fairly small wavelengths of light, fairly energetic. Questions here? Okay. Important to plants being able to harness this solar energy, they have things called pigments, and pigments are capable of absorbing different wavelengths of light. Typically, we're talking about visible light, so colored light that we can look at on a spectrum. That's what this figure is showing here. So we can see we have our visible light, and then this is showing within that spectrum of visible light, purple, red, and blue, or purple, and blue have shorter waves. They're more energetic. And then our red light is a little bit less energetic. They have bigger wavelengths. So different pigments absorb different wavelengths, and it is specific for the pigment. Wavelengths that pigments do not absorb get reflected. So when we think about the whole spectrum of visible light, pigments can absorb some, and what they do not absorb, they reflect, which is what this figure here is kind of showing. If you can piece out these different pigments, we have chlorophyll A, chlorophyll B, and then beta-carotene. So we could look at this chlorophyll A, for example. Now, these peaks are where the most is being absorbed. So you can look at this and say, okay, mostly like blue or teal light is being absorbed, and then also red. So that would be our chlorophyll. If blue and red are being absorbed, that means green is being reflected, which is what we see in plants. We tend to see plants as green because that's what's reflected. So chlorophyll is the major pigment in plants. It is critical for photosynthesis. This is how plants capture that solar energy. Plants have other pigments as well, specifically carotenoids. These can also be helpful in photosynthesis. Something that's really cool is they provide phytoprotection or photoprotection, where they help keep the plants from getting damaged by the sun's rays. If we look back at this figure over here, the tiny dotted line is looking at what types of light these carotenoids absorb. So you can see they're mostly absorbing blue and green, which makes them reflect red and orange light. It's here. So different pigments absorb different wavelengths of light. Okay. Now, as I was just talking about with those pigments that are able to help plants protect themselves against damage, this is related to why people say carrots are good for your eyes. So I have pictures of a whole bunch of different orange plants here. Why are carrots good for your eyes? It's the same reason that plants have those carotenoids to protect themselves. They exist in high quantities in some of these plants, like carrots, like papaya, like mangoes, sweet potatoes, etc. And the phytoprotective qualities that they have also apply to humans. So kind of cool. We can eat those plants that have those compounds, and then we also access those same protective qualities. We can't synthesize these pigments, so if we don't eat them, we do not have access to them. Photoprotective pigments specifically help dissipate excess light. So that's what they're able to do. That's how it works. That's the mechanism. Okay. And super important to this process are the specific tissues that this happens in. So leaves are really important organs. And when we look at leaves, we can kind of break it down here. So this first picture I'm showing you is, of course, a leaf. And then we can look at a cross-section of what leaves look like. We have this mesophyll layer, which is that inner layer. Then we have our cuticle and the different dermis layers. So the epidermis on both sides, both upper and lower. Then if we look even further inside this leaf tissue, we can see these really important organelles, which are our chloroplasts. This is where photosynthesis is occurring. So leaves are the major site of photosynthesis. This is where most of the photosynthesis is occurring. And we consider leaves to be organs because they're composed of multiple tissue layers. So that's our definition of an organ. So leaves do fit that definition. Now our chloroplasts are located within the mesophyll, which is that middle layer. So again, that's what I'm trying to point out here. And leaf structure is really optimized for this process of photosynthesis. The different characteristics that it has, including the vascular tissues such as the xylem and the phloem, which carry the products and the reactants of photosynthesis up and down from the roots. We also have structures called the stomata that are able to open and close at different times of the day in order to let carbon dioxide in and release oxygen out. So plants can kind of control when they do that to optimize this process. This figure is also pointing out the waxy cuticle that helps keep water inside of leaves and helps them reduce their water loss. Related to that idea of reducing water loss, that's why we see the stomata located on the underside of the leaves rather than on the top, because that helps protect them from the heat or the sun. And so they're losing less water when they open their stomata. Okay, questions so far? All right, I have a break for you. I don't have anything assigned for this break. I've given you lots of assignments. You can just rest your brain, talk to friends. It's your break. Give you like a minute or two here. Rest your hand. All right, ready to keep going? Okay. Here's our photosynthesis overview slide. So this is showing the overall process. You can see at the top here, and this is pulled straight from your textbook. We have the equation for photosynthesis. So carbon dioxide and water are our inputs. We also have to have sunlight. So that's important here, included in this equation. The end results are sugar and oxygen. So those are our products of photosynthesis. Humans definitely benefit from this process. We do need oxygen in order to fuel our own cellular reactions. So shout out to plants for helping out. Photosynthesis overall, of course, is capturing the solar energy and then bringing it to sugar. Relies on pigments we were just talking about to absorb photons. Now, photons are particles of radiant energy. So the sun emits radiant energy and photons are small particles, and pigments absorb those small particles. This process, as we were just talking about, occurs in the chloroplasts, and this happens in two stages. We have our light-dependent reactions and then the Calvin cycle. So the first part, as its name implies, needs light, but the second part can actually happen in the dark. It doesn't need sunlight for that to occur. So we have this broken down into two bits. You'll notice in this figure here, we have a plant cell. We can identify it by its large central vacuole. We have that cell wall. We have that square shape. Now, one of the things that makes photosynthesis such a significant process is not just the fact it's able to capture solar energy, but it's the fact that it's able to store solar energy in stable carbon-carbon compounds that can be used later. So it's able to both capture, convert, and store, which is really, really unique and cool. We're going to get into the light-dependent reactions, which is that first step here. So this occurs in the thylakoid, which I, again, have shown you on this figure here. You can see our thylakoids are being pointed to. They're like these little pancake-looking things. They're the little circles. So there's stacks of thylakoids in the chloroplasts. You can see those here, those little stacks. Those are all thylakoids. So there's lots of these that this is all happening in at the same time. So light photons, they hit chlorophyll pigments located in those specific organelles. Electrons are taken from water. That water is split, And then we get ATP and NADPH. And I'll talk a little bit more in detail about how this happens in the next couple slides. Now this equation is showing for the first step. So we start with light, we start with water, and this first step produces ATP, NADPH, and oxygen. Now oxygen is released as a byproduct that's not used in the next step. However, we know ATP is a useful molecule and NADPH is an electron carrier molecule, which is also useful in facilitating that next step of the process. So these products, ATP and NADPH, are used for the Calvin cycle. When the water gets split, oxygen is released. So that's where that oxygen comes from. Okay, so going into a bit more detail about how this happens, solar energy is captured in the photosystems. There are two photosystems, which I'll show you on the next slide. And this is kind of interesting here. So a photon of light strikes a pigment molecule. So that's what's being shown here with this squiggly yellow line. We have a photon of light. It strikes this green little pigment. However, electrons are not moving here. What's happening is actually that pigment gets taken to an excited state. As a result, that excited state is able to transfer energy. So this is an energy transfer. It's not an electron transfer. So this happens in the light harvesting complex, which is this big purple thing. Again, a complex is a group of proteins that are kind of hooked together to serve a purpose. So this energy is being transferred down through these different pigments. Ultimately, it's going to reach the center of this reaction complex, which is this lighter purple shade here. And then there's special pigment molecules that are able to actually transfer an electron. So these pigment molecules that are in the outside of this light harvesting complex, they're nothing special, but there are special pigments inside the center here. And those pigments are special because they can actually transfer an electron. So this electron gets transferred and then accepted by this primary electron acceptor. So this is how photosystems work. This is how they capture that chemical, or not that chemical, that solar energy. Questions here? Okay. There's two types of photosystems in the light-dependent reactions. We have photosystem 2, and we abbreviate that PS2, and photosystem 1. You might be thinking, what the heck, Nora? Put them in order. I did put them in order. PS2 acts first, so photosystem 2 is first, and then photosystem 1. Now this is confusing, I agree. It was named after the order in which they were discovered. So, I apologize, I didn't name them, or else I would have named them differently. But photosystem 2 acts first, then photosystem 1. So PS2 captures the light energy, extracts electrons from water. That's what its job is. And then that photosystem 1 happens next. It takes that electron and then is able to reduce this NADPH molecule. So they do different things, but they work in a similar way. Now this figure looks kind of scary, but it's actually pretty helpful in understanding our two photosystems and the way that they work collaboratively. So we can see our annotation here, PS2. So this is photosystem 2. This is going to work first. So first, in that second photosystem, energy from sunlight is used to power the breakdown of water. From that breakdown of water, we're able to extract electrons. Then oxygen is produced, which we can see over here. Water is split. So all of this is happening, released into the thylakoid. So that's kind of what's going on in the second photosystem here. We also have our photosystem 1. You can see right here, an electron from that second photosystem is going to get transferred to the first. It is also an electron transport chain. So similar to the electron transport chain that exists in the mitochondria, we have protein complexes embedded in the membrane of the thylakoids that facilitate this reaction as well. So as electrons travel across this transport chain, from photosystem 2 to photosystem 1. Hydrogen, again, hydrogen ions get pumped into the lumen. So we see hydrogen ions getting pumped into the lumen here. We once again have ATP synthase. So here's our lovely ATP synthase molecule. Same deal as cellular respiration. It works in the same way. Hydrogen ions are going to flow according to the concentration gradient through ATP synthase, starting that molecular rotor, and then being able to generate ATP as a result. So it's kind of similar to cellular respiration, just in this one part of the electron transport chain and the ATP synthase, but our protein complexes are different, but there are similarities. This is also happening, of course, in the thylakoids and the chloroplasts, not in the mitochondria. Questions here? Okay. So this is all for our light-dependent reaction. So this is all light-dependent. And then our products here are going to be used for the Calvin cycle. So we start with ATP and ATPH, made by our light reactions. Now, the Calvin cycle takes in atmospheric carbon dioxide. It takes in that CO2. And eventually, it's able to fix that atmospheric carbon and then also convert it into molecules that can be used to make sugar. This process is where carbon fixation occurs. So if you've ever heard about that in other like environmental biology articles or anything, when they say, oh, the ocean is really good at fixing carbon, it's because there's organisms that are able to capture atmospheric carbon via photosynthesis, harness it in their tissues, in their bodies and things, and keep it from going back into the atmosphere. So this is where carbon fixation occurs. The Calvin cycle does not require light. It's just requiring the products from the light-dependent reactions. And again, I have put the equation up here for you for the second part of this reaction. So we're starting with our inputs, ATP, NADH, carbon dioxide, and then we get sugar. Now, I didn't put just glucose here because the exact sugar molecule that's formed is not glucose. So we'll talk a little bit about that later in the next couple of slides. So this is our overview here of the Calvin cycle. Now there's 3 steps to the Calvin cycle. We have our carbon fixation step, we have the reduction step, and then regeneration. So the carbon fixation is where this specific RUBP molecule accepts carbon dioxide and then is able to make something called 3-PGA. The name of that is just kind of silly. It's not making 3-PGA. The molecule is called 3. So that's its name. Now, the enzyme Rubisco is really important to this process. So that's what's showing right here. So the enzyme Rubisco helps that along. That next step is a reduction. So that's where ATP and NADPH come into play. They're going to take that 3-PGA molecule and turn it into G3P. Again, a silly named molecule, kind of hard to remember. But so you can see these stages. We first have that carbon being fixed. Rubisco helps take it to that second stage, the reduction phase here. And then our final stage is the regeneration. So some of the G3P molecules leaves to make other things like glucose. Maybe it's going to make starch. Maybe it's going to make cellulose. It can leave and do other things. Some of the G3P that's produced is going to go back into this cycle. So some stays and some goes. One carbon dioxide molecule is used in one turn of this cycle. So to make a glucose, we would have to have 6 cycles. To make one glucose molecule, this would have to turn this would have to turn 6 times. So I mentioned here for this regeneration step, which is where we're regenerating this RUBP molecule. The reason we need to do that is so that the cycle can continue to turn in order to make that glucose molecule or those other sugar molecules. So Rubisco is what we start with. It's also what we end with, which is how these cycles tend to work. Okay. Realizing we're out of time. So we're going to stop here. We'll review at the start of class next time.